Welcome, everybody, to The Soil Matters. Av, this is a friend of yours, so I'm just going to jump back into the background and let you introduce him.
Thanks,
wrong
Ken.
button.
Oops. Thanks, Ken. Nice seeing you, Leighton. And yeah, this is a real privilege because every time I've invited Claude to be on a webinar or something like this, it's usually only for like 10, 15 minutes, 20 minutes. It's on a panel. And if any of you have met Claude, 10, 15 minutes, he can just tell you about one particular life cycle stage of a particular beneficial insect. You know, if I was looking for an OG of Eastern Canada, Claude Robert would definitely be one of the OGs. A phenomenal lover of cannabis. Advocate for cannabis, despite the fact that his province does not allow you to grow their own medicine at home. But that was just a cheap shot to Quebec government. And but Claude comes with an incredible wealth of knowledge around creating a healthier environment for growing your crops. He's the I call him the technical sales director of. He's got a more important title, I'm sure. But working with Anatis Bioprotection, absolutely love the products that they sell. It's not only selling of the product; it's actually the knowledge around what products that are best to be used, and actually rearing a lot of their own insects and being able to say, "Hey, this is coming fresh off the farm. It's going to do amazing stuff in your garden." So, with that, I'll turn it right over to you, Claude, to introduce yourself more properly, but it is quite an honor to have you here. Thanks for coming.
Thank you, Av. It's always a pleasure to be around you at all the events, or virtual or in person. So that's it. I've been passionate about biology, focusing on agronomy, entomology. I love cannabis. I grew my first plants in '86. Back then, in the days, they called me the druid because nobody ever seen buds before, because they were all like, before that, the equatorial varieties, they would grow, try to grow in Quebec, and they would never flower on time, you know, so they would call it Quebecois, so it was weak weed. And then the first seeds started to appear from the seed banks in Amsterdam, from the travelers that went around in the Himalayas, or there's South African sativas, things like that, the Durbans and things like that, that finally could flower. So I've been involved in that part of botany for a while. I also love a lot wild plants, identifying them. I also worked in greenhouse research since 2004. I worked for the research project for the greenhouse growers of Quebec. And I also taught fruit production. I was also an advisor for small fruit producers, and also Chinese vegetables, pak choy, bok choy, all the giant radishes, the Chinese and Korean and things like that was fun. But I was not having that much fun because I was stuck in the conventional side of things, you know. Mr. Smith, we reached the threshold for tarnished plant bug. You have to apply ripcord and kill everything in your field for weeks. So I didn't sleep well at night. And so my father told me, "Hey, there's an opening at that Anatis Bioprotection. Did you hear about them?" "Yeah, I heard about them." Yeah. I went there. I said, "Oh, yes, I'm really at a good place with the good guys." No, the good girls. We're all girls, bug nerds back in the days. Now we're 7 or 8 employees. Now we're about more than 30 people working at Anatis. And we rear our own beneficial insects. What we don't do, we're partnered with Applied Bionomics in BC, in North Saanich on Vancouver Island, and they reared some of our stars, Anatis baccharum, the crazy mite, Gylolelaps gillespii, which is the cousin of Stratolelaps simitus, also known as Hippoaspis miles. So we do that. One part of Anatis is the predatory mites and insects, and we also have nematodes. But the other part is the microbiology department. This is situated in Laval, in the National Research Council building. That's where they do the quality testing for our own Bavaria strain, Borrelia bassiana strain, and 03. That's what started, in fact, Anatis. It was our founder that found a spore on the tarnished plant bug in Quebec. Then she identified it, did all the registration to say, it's your strain, you found it, then you multiply it, you bring the product to market, you do all the efficacy trials that you have to go through in Canada. We're now in the States too. We're very efficient again with cannabis aphid. I just had another report of a real protocol, like a real research protocol, not just bro science. We weren't at 100% because 100% is not possible, but we got them all. We do it, the application of the Bavaria, we do it during the propagation stage when the relative humidity is really high.
And Claude, maybe we'll jump into that very specifically when we talk a little bit more about some of the major problem areas that you get to see. Both, I'm hoping that we can both address some of the stuff that you might see in hydro gardens versus living soil gardens, and greenhouses versus warehouses versus outdoors as well. And I think a lot of our audience does like to grow outdoors, and we're getting to that season, so hopefully we can talk a little bit about that. But yeah, definitely we want to hear a lot more about the cannabis aphid and better protocols around that. But yeah, with that, I also want to just introduce, of course, our host. You two probably know each other from the Future Cannabis Project, but Leighton, do you want to jump in and?
Oh, I can't wait to hear more. The cannabis aphid was like, just coming around about 5 years ago. People were talking about this new aphid that was attacking first hemp, and then it had worked its way to some of the more traditional, THC-based cannabis. So yeah, please dive right into this, because this was always one that was really like scary. Like, whoa, what is this?
And ugly, incredibly.
So for Oregon cannabis, the cannabis aphid, or the bang aphid, BHANG, comes from India originally. It's been traveling around the world following people that plant cannabis. It appeared, the legend says, I don't know if it's just folklore or urban legend, that it was in 2016 in Oregon that there was a convention, people were meeting, and this guy had elite cuts, all sorts, and he gifted people with these cuttings, and they all went through America with it. So because it's usually the way that people transmit it, it's always through cuttings, always. Now, on the West Coast, it's not the case anymore. And in Ontario, now it started, and I believe it started also in BC. They started to be endemic, meaning that they survived, and now they're part of the landscape for outdoors, meaning outdoor growers. For indoors, it's always brought in by, very rarely, I would say, but there's in fact, there's 6, or maybe now we're down to 7 species with aphids that have been identified that attack cannabis. And one is the cannabis aphid is very specific. It won't eat hops, it won't eat pepper, it won't eat just cannabis or hemp, cannabis, whatever. So, but the other aphids, they attack different plants, like the peach aphid, the melon aphid, the dandelion aphid, the bean aphid. These all will attack cannabis, and the foxglove aphid too, but not the cannabis aphid can only be found on cannabis. So if you get cannabis aphid, you got it from a nearby field that's very close to your facility or to your field, or you got it through cuttings. And usually cuttings, they have to be quarantined, and people don't quarantine enough or long enough. There should be 21 days minimum, or bring it up the temperature. Be sure, because I've seen people that really clean facilities, and then they wanted new strains, and we're really, let's get this strain in, and you got a new friend in your building.
So what else do you want folks to do if they're going to quarantine? They're bringing in some new cuts. You want 21 days. Are there other things that you want to treat those cuttings with?
But I'm getting the most extreme and the most like 21 days. But I mean, you could dip also your cuttings in the oil. If you have to use an oil. At first, when I first learned about oils years ago, I thought vegetable oil would be better, but no, finally it's mineral oil. That's good, nicely refined. Soft oil X is the one I would use. We have an EC formula of our Bavaria, the Bioceres EC. It's approved by Health Canada for cannabis production, the WP, the wettable powder, and the EC, the oil-based emulsifiable concentrate. They're both certified organic by EcoCert. They're both listed on MRI. The others are not because they got synthetic.
And you can dip with Bioceres?
Not according to label yet. That's what I was about to say. Okay. So people that are doing have to follow strict rules. They go by the strict rules. The people that don't have to, they do what they need to do. Some people were even cheating, I would say. They were doing a sprench instead of a spray or a drench. It was a sprench. But we finished our the papers being written right now for submission to PMRA this year. We're waiting for the cold fogger application. I'm waiting for outdoor application, and we're waiting also. We're on the works too for a new granular. The granular will be put in the soil to get the pupa of thrips and other pests. So it was mainly for Japanese beetle and rose chafer and a cinch bug for lawn, for turf. But now it's been, we thought that, hey, let's use it for trips in the soil. So it's just going to be a new tool in my pitchfork approaches for like the time release nematodes and alginate capsules too are coming soon. So this is going to be exciting.
Is there a predatory bug for those cannabis aphids, or is it just the surfactants that you think are?
You have to choose, like you have to use it. In fact, I would say a combination in your pitchfork approach. In fact, a pitchfork approach would be to use as many beneficial BCAs, biological control agents together that are compatible to fight these nasty pests, you know, the ones that are really tough, like the cannabis aphid, because usually aphids, all the other species of aphids that attack cannabis will have 4 babies a day when the aphid is adult, but the cannabis aphid, don't say cannabis doesn't make you productive. They can produce 6 babies a day. So it goes even faster with cannabis aphid. And as hotter it gets, the more and also the influence by the photo period of the 12-hour photo period, they think it is fall. So they produce eggs. We haven't been able to find, there's not been enough research yet done for many reasons to see if these eggs could be able to hatch or they need a cold period, you know. If they need a freezing period, like a winter or something, to be able to hatch. Because we haven't seen that kind of cycle happen. But just one aphid is enough to restart everything. Because usually they will just reproduce asexually. So meaning the mama is giving birth to babies that are pregnant, of babies that are pregnant. So they just continue on that way. And usually in nature, like during the fall, there will be some male individual that will appear, and there'll be a sexual reproduction, meaning that then they'll lay eggs. But and they also produce flyers all the time, so you have to watch the flyers. Flyers happen when the colony is getting too big. They have a stress, sprays, the photo period change, some things we don't explain yet that makes them build like have winged individuals that will fly off and try to find colonies elsewhere.
So if you had an outdoor crop last year and you had a cannabis aphid infestation, what can you be doing now to try to prevent getting it as bad this year?
You start by planting your islands of biodiversity, meaning you want a lot of sunflower, marigold, alyssum, lots of alyssum, things like that. Things are going to benefit the predators. They're going to furnish them a lot of food and fuel, nectar and all that, pollen. And so you start by that. And also you will introduce the aphid predator image. The aphid predator image, less is more, meaning you don't have to overpopulate your area. They're native from Canada. So it's very smart proof. It's a max vial, we call it. It's like a big pill bottle. Inside there's some substrate, some pupa of Aphidoletes aphidimisa, the aphid predator image. And this midge is very special. It's a big midge. And it also loves to stand on spider webs. It doesn't stick in them. Just as an observation, some point, and it will go and lay eggs next to the aphid colonies. And it's its larvae, it's bright orange, that will eat aphids like crazy. And they're very furtive, and they like to hide under aphids, and they like to work at night, at sundown. So when you scout them, you have to move the aphids to see that. If they're established, they're under the aphids, but meaning it will be very easy. You just need 250 an acre, and it's about 20 something bucks, and you release them a couple of weeks, maybe I would say 3-4 weeks to be sure that your population is established in your area, and that way you should be able to manage your aphids.
Would you start like May 1st or?
Depending on where you are.
Yeah.
In BC, West Coast, I would start as early as possible.
Okay.
And in Canada, and I mean on the East Coast for us, and it's even from region to region, from north to south, it's different. But I mean, you try to follow. You dig in some information from your area, or you remember your history when they appeared the year before. But also, I would release them as soon as possible in the spring, when it's warm enough. So make sure, because you always make sure that when you introduce, you're not introducing too late. It's not bad to introduce too early, because you don't want to miss it. Like us, in garlic, we have the swin midge, and things like that in garlic and leeks. And if you don't introduce too early, then it tends to be earlier every year because of all we know, climate change or global warming, things like that. You might miss it. It's too late. It turned into a larvae, and now the trichogramma that we released to go parasitize the eggs cannot get the egg. They're already hatched, so you're done. It's too late. The same thing with the corn borer in the cannabis outside. When we released the trichogramma, so we rear 4 species of trichogramma wasps. They're very tiny. They're like a point on. I don't know. They're like one of the numbers there. I know it's one, and it's very, very small. And 4,000 pupa old, like in the size of $1. And we used to glue them on cards. Now we have a new release method. It's a little biodegradable cellulose capsules that we either disperse by hand in the field, or we load a drone. We send a drone that drops a capsule every 100 square meter, or 1,000 square feet for our American listener. And these wasps, they emerge, and their only goal in life is to find eggs of caterpillars to go and parasitize. And we have got specific species that will parasitize either the borers or this type of caterpillar. So we're even better than BTK. Because BTK, you have to wait for the right time to spray, and it's non-discriminate. It kills every Lepidoptera, whatever, butterfly. Us, we target the one we want with the trichogramma, So we get with the trichogramma austrinia, we get the corn borer. With the trichogramma brassica, we get the corn earworm and other types of and we even have trichogrammas against a flower moth and also the ones clothing moths that so the ones that attack grain or clothing and animal fiber clothing. So we have little kits for the house. Instead of putting chemicals in your house, you just release them. You don't even see them, so you're not bothered. And they will find the eggs that they need to parasitize. Our new website is we're testing it this week. I liked our old website, but our new website is going to be so much better. Thanks for showing it, but I'm not proud of your old website. It's too slow. Anyways, the new website is going to be online very, very soon. It's going to be amazing.
So let me ask you this, Claude. Have you noticed any difference in really, really biologically active, healthy soil systems versus the less so or conventional ag type scenarios with these pests?
Exactly. Just to give you an example, we sell predatory mites. We have 2 species, Stratolaps simitus and Galolaps gillespii. 2 are predator mites that live in the soil. And the customers that are using like Pro Mix, whatever, or just hydroponics or Rockwool, or they have a bit of trouble keeping them alive. Like with Galolaps, it goes well because they are surface dweller and they can live on compact substrate like cocoa blocks or slabs or Rockwool. But Stratolaps has difficulty. Nematodes, they get expulsed because they have no organic matter to cling on. So using non-organic methods doesn't help our predators or organic predators. You know that they're not as efficient in these type of soil. I mean of substrates. So I would say for sure when the plants, we are just like doctors to help the people that get unhealthy. Because if everybody was doing permaculture and would have a biodiversity islands, clean cutting, good methods, good spirit, good energy all the time, nothing would happen. The plants would defend themselves. And there was no monoculture and no, then there would be enough predators and everybody around to cope with things, But now, even in cannabis, we're doing monocultures of cannabis. So what I've been pushing a lot, and finally we got our letters from Health Canada telling us that we can use non-cannabis plants in cannabis production facilities to help with IPM programs. Because all these QAs, all these quality assurance people, they all came from when the industry got legal in 2018, really big time for recreational. Because before that, there was just organogram or canopy back in the days for medicinal purposes. But when in 2018 it started, they needed to hire all these quality people, and they all came from the pharma sector, food processing sector. So they had no idea what they were doing, and they were overcautious for some reason and some not. Anyways, and they never permitted any other plant than cannabis to be grown in these lab sterilized facilities, So then I started to visit my first organic growers, like the Green Organic Dutchman. They were doing indicator plants. They were growing bean plants to catch spider mites, to see if they had spider mites right away. And the beans would feed the kitchen, the employee's kitchen, because you're not allowed, and for a reason, I respect that rule, is that to sell what you produce in the cannabis facility. So they wouldn't be able to have a road stand on their beans next to the road. But so that, then Fuga in Quebec, she was doing cover crop plants on her living soil. So then they can tolerate that. Okay. Then I realized, sorry, I have a singing cat. It's going to come entertain us, so I had to block him. He's very special. I think I'm going to make him a YouTube star soon. But a white cat that sings. So where was I?
We're at Fuga.
Oh yeah, okay. Yeah, so the cover crop. So then the first, because we've been producing for years, we do aphid banker plants. So we grow barley in a small tray, and in this tray we inoculate them, we infest them with some cherry bird oat aphids. So these type of aphids, they're very tiny. They just attack monocots. They just attack monocotyledon. So they just, unless you're producing chives or you have ornamental grasses, you don't have to worry about them. They won't go attack other types of plant. So they serve, in fact, as a breeding farm for our parasitoids of aphids. Aphidius colemani, Aphidius cervi, Aphidius matricariae, which is the best one for cannabis aphid. These aphid parasitoids, they will go and find aphids, pierce them with their ovipositor, and insert them an egg. So their larvae can eat them out, inside out, and they will eat, like in the movie Alien, they will eat their way out of the aphid. The aphid turns into a big mummy, we call it, like it puffs, it turns grayish, silverish, we call them mummies. So when we sell these parasitoids, you receive a small tube, and it's filled with dead mummies, dead aphids that are all parasitized, and they will emerge in a day or two when you release them in your garden, and they will try to go and find aphids to parasitize. And when you want to install them, like with a banker plant system, so that way they're present if aphids show up in your greenhouse. They're already living on the aphids on the banker plants. And in fact, what we sell you is a starter. So you sow your own barley, oats, wheat, winter wheat, whatever. Barley is the best. And then you just maintain your plants. You just sow some new ones after a couple of weeks, and you just stick the tray we sent you next to your own tray, and they will transfer to the other, and you just keep on going with that. So that way you release your aphidius, and that way they will be present in your greenhouse or your indoor garden.
So if you used a monocot as a cover crop, would it double as a banker plant, or?
Exactly. Yeah, exactly. You could do it that way, because these aphids, they might be, if you know what you're doing, I mean, if the people know it's no danger, they have no worries for their cannabis plants or tomatoes or whatever other type of plant, the other dicots. There's no worries there. We also started talking about banker plants, ornamental peppers, purple flash. So these will serve for food for Aureus, which is a really good trips predator, and also for other, and they will reproduce in these ornamental peppers. And with tomato growers, we use mullein for Dicyphus, another predator. We also have alyssum, that's for outdoor growers though. Indoors and in greenhouse for cannabis, there's petal pollution, meaning you don't want the little white petal going to stick in your flowers of the alyssum, so we don't use them indoors or in greenhouse. Same with the Aureus I was talking about, this predator of aphids, not aphid, trips, is actually a little bit of young aphids though. But the Aureus, it go sleep at 12-hour cycles, so you never use them during flowering, they're lazy, and they will go stick in the flowers. So usually I only use like the ornamental peppers for cannabis growers. I would use it in their mother rooms within veg areas, and that's it. Never anymore in the flowering. The other companies might forget to mention that, but I like to tell people that's how it works. So
yeah. So just to repeat that, for you, Aureus is something that you only use in veg. Yeah,
veg, mother plants. It's a very good predator. You know, it hunts for sport. It will kill many trips a day. It flies to the trips. When you have the pepper plants, it can feed itself on pollen and reproduce easily. So it maintains itself. You always see new nymphs. And the same thing with the Stratolelaps and the Galulaps. When you have living soil, usually you introduce them once, and they go on by themselves. And when the people reuse their soil, they're still there in the soil. So they take care of fungus gnat, shore flies, pupa of thrips, pre-pupa of thrips, and other soil-dwelling pests or allies. But that can, I'm talking now about springtail, Columbola. We receive so many pictures every week of people, hey, I got root aphids, or what is that? Wow, it's freaking out, whatever. People with cocoa, people with living soil, usually, or just soil, but I mean living soil. Yeah, sometimes there will be a lot of Columbola, a lot of springtails. There's a couple of species, some are whitish, some are translucent, some are blackish, some really like spring, some are less springy. There's many species, and they're soil decomposers. They're your allies. They're working on your soil. But if they get too many of them, they stress the plant because they start to feed on the roots, and the plant, instead of producing flowers and fruits and leaf, it concentrates on always repairing its roots. I mean, so it's always repairing its roots.
Leighton, have you ever seen that, where springtails dominate to such a point that you start seeing some damage to plant roots?
Only in really, really overwatered situations. And that's usually the biggest problem, is that people are overwatering, and then, yes, they bloom like crazy. Now, what about the rove beetle? Do you ever have, is there something that people can do with rove beetles? Because they're the same way. If they get friggin' out of control, they can create a real bad imbalance.
Exactly. Because we have them, the rove beetles, they can be very efficient in certain situations. I even had the stories of some of our customers that had a problem with a lot of fungus gnats and shore flies coming out from drains. So they would dump the Delosia in the drain. People with aeroponic system that were struggling with some sort of fungus gnat because they had so many mats of roots, that was crazy. It would work well. But Delosia is very tricky because they're so generalist, and they will eat whatever they find. So many times they go and destroy all the slow release sachets we have for predatory mites. They go and pierce them, they go inside. So they're as destructive a bit like your wigs will be, because they're fantastic predators, in fact, in a way, but they're they're too general. You know, it's like I would compare them like praying mantises, or they won't cure your problem. They will just eat here and there, and let's trash this, and let's eat that, and let's go swim, and then let's dig in the ground, and then let's whatever. You know, it's like some people really love them, and I fill their order when they ask me, oh, I want to buy some. Okay, let's go. But usually, since they're not compatible with they're not really compatible with the Bovaria too. Aureus and Delosia have trouble with our strain of Bovaria, and also because the way we don't have these weird material sachet. We have the paper one, so our sachets are not the Delosia and their sachet are not really. They don't like each other. So that's one of the reasons it's not my favorite predator, but it is a good predator in certain situation. It can, for shore flies, it's amazing. For shore flies, it's the best way when you have a lot of algae. Delosia, Correaria, Teta, the old name, the rove beetle, is for a big shore fly problem, they're the number one. But for other things.
Have you heard that dry dog food can attract them, and you can collect them that way? Putting little bits of dry dog food and just collecting them and getting them out.
Okay. And there's many, okay, to get them out of the earth.
If you've got an overpopulation, put some bowls of dry dog food, and they'll go to that area to collect them.
Another trick is cornmeal. For whatever reason, they really love cornmeal.
Maybe that's in the dry dog food.
It probably is, right? There isn't cornmeal.
I really loved your show, by the way, with the fish brew. Oh, wow. You really taught me something. And, you know, I just knew about hydrolysis stuff. And like, I said, wow, why? Yeah, you cannot know everything. Nobody's omniscient. That's why we have to share and teach everyone of everything we know with a passion. And because I really learned, when is this stuff coming? Anyways, we'll have to talk because we maybe want to distribute that in Canada, that fish brew thing.
Oh my God, that stuff is just amazing. I have yet to not see incredible plant reaction within 30 days. And I'm talking on redwoods, 400-foot tall redwoods. Within 30 days, I saw plant reaction. The stuff is gold. And I was talking with Keith today, and I asked him, how you doing in Canada? He goes, dude, it's just a fucking nightmare. So anything that you guys can do behind the scenes and then relay it to him or to me so that we can hit it from both sides, the quicker we can do this. But I mean, you guys are like so close on the East Coast. He's already, he's only like 4 hours away from the border. So it would be nothing to transport it to you guys. But
because we're already, we'll talk about that fair. But we're doing business with the USA now. We are selling to some beneficial insect providers in the US. We know about how to go with the registration and things like that, and to get things into Canada. So yes,
that's amazing. That sounds great. So yeah, you guys, you keep the pressure on that side, and we'll keep the pressure on this side.
We're working right now because Anatis is just, for now, it was just a BCA, biological control agents. So meaning we'd have predatory mites, predator insects, parasitoids, nematodes, and the microbiological, like the myco insecticide, the Bovaria massiana and 03 strain, different formulations. And we're working on a new one for flies soon, against the cabbage fly and things like that. Very soon, but very soon. In this world, it takes years, but soon. But we're on the right track. And we also now, I want to market though. There's Urtalive that's organic. We distribute that. It's some kinds of bacillus for the soil. I've just started to do that, and I'm preparing also some 2 types of insect frass. So some black soldier fly and some mealworm frass from 2 companies in Quebec that produce protein, and they want to market their frass. So we might do that because it's related. We want to do I don't want to be like plant products and sell fertilizers, but I want to do related products that are in the frass from insects or even fish brew, because I know it's living, it's a living product.
So Claude, let me ask you this: would it be easier to bring it in as a biological? No, that would be worse.
No, it's worse. You have to stick with the fertilizer. It is a fertilizer.
Yes. Okay. All right. Fair enough. Figure that out.
And there's nothing living in it. It's just a fertilizer.
Yeah, they kept alive. It's very funny. Neem oil is not permitted in Canada for different reasons, weird reasons. And as a plant luster, though it is, to shine the leaves of your plant, make them like.
I don't think you're allowed to even use it as a leaf shine anymore.
Okay.
Yeah.
Yeah, but back in the days, it was just.
Back in the day. Great.
No.
Yeah, it's a challenge.
So some of our predators, they cannot be sold in the States because they haven't found them yet in the States, but we know they're living there. Like the crazy mite, Anatis macaron, the new hyper predator, the big and orange that eats aphids, thrips, white flies, spider mites. These crazy mites we cannot ship to the States. We cannot ship Galolabs, Gillespie, the cousin of Stratulat, because we're sure it's on Washington or Oregon or North California, but they haven't found it, so we are not able to ship them there. All of our beneficials, they are all, most of them are native species. The ones that we have that are not native species are non-invasive one. that been allowed.
Yeah.
Like Persimilis, which is the most known for spider mite control. Phytocilius persimilis. So these little guys come from Chile, and they Swirsky, the most known predator for trips because of our big competitor. Anyways, these guys come from the Mediterranean area, so they come from there, but they wouldn't be able to reproduce and live and invade Canada, let's say, or that's why they're allowed in greenhouses and door facilities, and you can use them there. But most of our species are native, you know. Like we have one predator mite that's called Neoxylus fallacis, and I sell them to raspberry growers and strawberry growers. And for the raspberry growers, it's just a one-time introduction. It's going to live on year after year in the field, but it's native, so it takes care of brown mites and spider mites.
Claude, I was wondering if do you have a video of the crazy mite that Ken might be able to pull up, or?
Yeah, I mean, there's one that because I have it on my computer, but I cannot.
Sorry, it's not. I was wondering if it was on your website. Leighton,
No.
I don't know if you've seen the crazy mite.
Not yet.
No, I haven't. Is this something that was bred, or is it just something that popped out of nature?
So Anatis Bioprotection, aka the whirligig mite, aka with the trademark name, the crazy mite with two E's.
C-R-A-Z-E-E or
Yeah,
C-R-A-Z-E-E.
Yeah, so Anatis Bioprotection was rediscovered, in fact, by Taro Saito, a researcher working at the Vineland Research Station in Ontario. That's an agricultural research station that works a lot on biological control. And he was walking in the field with his son, teaching him what nature is. This is a bee, this is a flower, and what is this, Dad? And Dad looks, and at the same time, it sees that big orange predatory spider jump on a fly or something like that. So he said, "Oh, you're coming with me." So we captured it, brought it back to the lab, and they've been working with it for years. Before they were able, with a lot of research, to find a way to rear them commercially. Because they'll be cannibalistic tendencies. So these type of species that have cannibalistic tendencies, it's an extra challenge to try to rear them in a way that's efficient and you can produce as many as you want, scaling it up. And so we found a way to rear them commercially, and now it's been almost a year. We've been distributing them across Canada, and we're still learning about it all the time. We know they're able to jump on psyllid, on trips, all species of trips, all stages, on fungus gnat adults, on shorefly adults, on spider mites. They're able to walk across spider mite webs. They're able to walk on trichomes, capitate stalk trichomes in cannabis. They don't stick in the flowers. They're able to walk across them when the people come into the rooms for harvest. You see them falling off the plant, running on the floor, on the tables. The complaints we had is people that are really nervous around insects. Some ladies at the reception or whatever, going, "Oh, wow, they're everywhere." Because so, yes.
I got to assume it's a webless spider, correct?
Exactly. It's yeah, exactly. It's a strategy, in fact, in life, is to run erratically as fast as possible in a zigzag pattern until it can bump into a prey and it can embrace it. And then it can embrace it, it eats it. But lately,
how big are they?
They're 10 times the size of a Swirsky, I would say.
Okay, so big enough, obviously, that you can see them, but not so big that they're scary, right?
No, I mean, some people, they makes them nervous, yeah.
They're non-poisonous, right? They're not going to bite you.
Yes, they can a bit.
Oh, they can bite you?
Yeah, we're still in research right now because Taro, the people that are rearing them, they sometimes when they had hundreds on their arm, they had little allergic reactions, little pinpricks, nothing bad. We had one customer complaint. on.
Because that was what I was going to ask. Is it itchy? Like, are they, they get a welt?
Yeah, because what we are investigating and they're doing research on is to find out, are they really venomous? Because they might be venomous, they might be biting like a spider. Because Tarot, while he was doing research for all these years, many times when he would put a prey in with the anesthetist, he would observe them, and sometimes he would see anesthetist trying to embrace the prey, not being able, the prey would escape, but then the prey would die a minute or two after. What happened? So they're working on the research about that, and the way when he had hundreds on his arm, the way he got bitten, I presuppose that there was some sort of little venom they would use to hunt their prey. But besides that, all of our predatory mites or insects, there are no danger for humans or pets, and they won't become a problem for your plants. There's illustration of these crazy mites.
So, wow, this is like a super predator.
Oh, yes.
It's taken on almost all of the different pests. That's amazing.
And now we found a way. Sometimes it wouldn't work at a greenhouse facility or and we would wonder why. Finally, with lots of observation and gathering information and everything, we realized, and Tarot too, at the same time, everyone at the same, on their part, our friends at Applied Biodynamics, they're the ones who are mass producing them right now. They won the contest. They found out how to do it with Tarot, so they're the ones. But we have exclusive distribution, whatever, with across Canada. But meaning that when they found out how to read them, so they would work everywhere amazingly. We're still investigating if they could be used as a standalone. But they run so fast, that's crazy. And they are so easy to scout. So let's say for aphids, we put them when aphids, they do wonders. If they team up with something. Because the anesthetists, they will go for all the three first instars of the aphid, not the last one, usually. Or the too big, the ones that are too big, they really won't go for the last instar. So what they will do instead, they stand up next to mommy, waiting for her to pop her babies. They get them. So there's also a lot of their how they help is that they scare all the pests. They arrive and the pests leave, or they're always disturbed. They can never eat in peace, or reproduce in peace, or whatever. They're always stressed because they're always there and running. And hey, now there's a crazy mite coming. Let's run. So it's always the same. So especially with whiteflies, it's amazing what they did. They jump on the adults, and they scare off the rest. So they're never able to do their thing, So
wow, this is amazing. They even do whiteflies?
Yes, they jump on adults. So if you can find Taro Saito, there's a page on LinkedIn. He has a video on his website. If you could show that, they go after an aphid, or I have it posted on.
Can you type in his name in the chat?
Yeah, I have it posted on my LinkedIn too.
Yeah, well, do you mind typing in Taro's name in the chat?
Yes. And so the only time it wouldn't work is that they wouldn't wander aimlessly across the facilities, because they're the easiest also to disperse. You just open the bag or the container, let them go. And they will go from one end to the greenhouse to the other in lightning speed. The other ones before were tedious to Persimilis. You had to go one plant to the other, tick, 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 with a small little vial. Here's a little Persimilis here, a little Persimilis there, a little Persimilis there. But and it's just you open up the bag container. Let's go. Let them go. So when there was not enough prey, they would just go around the place, on the roof, on the floor, everywhere. But now what we do is that we introduce another predatory mite, Neozydius cucumeris. And yeah, this is a good video, but it's not the best one. You have to check the other one after that. But you can see how fast it runs. But if you go in his post that he did earlier, you're going to find the video against the whitefly, the aphid, and the trips. And so what we do is that when we introduce them, we will also introduce some bulk cucumeris. The cucumeris, they come on a substrate of a bran. And so we do little piles of this bran next to where we're going to release Anatis. So Anatis has a snack when he comes out. So cucumeris will eat trips, will eat brown mites and eggs of spider mites, but it will also serve as a food source for Anatis. So we do the Anatis cucumeris combo, and it goes like they stay there, they stay put, they have food. So we have slow release sachets of cucumeris and little piles of bulk that are available right away for them to eat. And
wow, this is a hell of a breakthrough, my friend. Yeah. This is amazing.
The last picture you showed, it was a new species, but it's not a new species, but it's a new species in Canada, the Parvispinus trips. It's the worst trips we've ever seen. It's black. It's really bad. It destroys your plant big time, fast. They came with ornamental plants from Florida. They were shipped to Canadian producers. A gift from Sunshine State. Yeah.
How is it they can ship fucking plants loaded with pests, but you can't get fish brew, right?
Exactly. We cannot send them the Anatis to get
fuck, man.
But they can send us the pest they have here.
Well, you know, I've got a few questions. I'm just going to throw them out all at once. I'd love for you to talk a little bit more about your biodiversity islands, and perhaps talk about how many you might put into, you know, in a backyard, how big you want it. If you're growing like an acre, how many of those that you need. Next, I'd love for you to talk a little bit about some foliars, whether it's insecticidal soap or bioceres or lactic acid, something, you know, just talking about things like what you might be doing for PM. And well, I think I had a third question in there, but I'm sure I'll come up with that. Just want to throw those out there, however you want to tackle anything.
Okay. For sure, with all the biodiversity islands, and the more, the better. So you do what you can, but you tend to favor the ones that are more productive. Like that's why I was mentioning some sunflowers, things like that. So you want to have as much as you want. And like I said before, also like alyssum you won't put in your greenhouse with cannabis, because you're going to have these tiny wet petals stuck in your flowers everywhere. So you have to watch that. Sprays, I tend to prefer sprays for prevention, for early plants, for cuttings, for propagation, for early veg. For sure with cannabis, I prefer not to, and I don't recommend to spray unless you really have to during flowering, because you're going to damage trichomes. You're going to leave some residue in the flower, either an oil or some adjuvant or something like that in your flowers. So for outdoors, what's approved for cannabis outdoors, we distribute one of these products. It's called Cyclone. It's also called Tivano. It's a citric and lactic acid preparation. It's produced in Quebec. It's approved by Health Canada for cannabis production and with many crops. It is a bit corrosive because it's a bit strong with the citric and lactic acid for cannabis, for the bacteria, the pistils, and the trichomes. So what I do is that we do a tank mix with a garlic extract they have. That's called Influence LC. It's also approved for cannabis production. And we do a tank mix of both, but a much lower concentration of each. So instead of 12 milliliters a liter of Cyclone, I would put 3. And instead of 16 milliliters a liter of Influence LC, I would put 4. So that way it makes it a lot less corrosive. You don't hurt your bacteria, your pistils, or your trichomes. And it's also the synergy of both make it 3 times more efficient. And it costs you 25% less. So that's a big licensed producer that told me that. They did their things. And like Lydie, I love bro science too, but there was also something about real science, real protocol.
Man, where's all the gold bars, motherfucker? That was huge. What he just fucking described.
Okay, I'm in the background trying to find his post on his LinkedIn account, okay? I'm working here, man. I'm working.
I think you need to repeat that recipe, though, or get someone to pop it up on the chat, because I think that's really, really important.
You're going to find it on the label of Influence LC.
Okay.
It's a garlic extract, 15% garlic extract. It doesn't give a garlic taste to your plants. It's certified organic 2 product. And it's also the Cyclone 2 certified organic, Tivano, and they all are approved for cannabis production.
And you can run them right into flour.
If you're using less, the recipe I told you.
And all the way through flour?
I mean, trichomes are trichomes unless you're using cold fogger. Like a cold fogger, because any spray will damage, even if it's just plain water. But it makes it less corrosive, because the Cyclone by itself was very corrosive for cannabis, meaning there was earth would have some tiny phytotoxicity and some pistil burn and some bacteria burn, and for sure would damage some trichomes because the citric and lactic acid are acidic and at the full strength. But when you do the tank mix, the synergy that happens, and it costs you less and it's more efficient. It was 3 times as more efficient for powder mildew, because for powder mildew you want to use something too. Sometimes because it's spreading so fast, sometimes you want to really stop it, and so you almost have no choice. People love regalia, an extract of Rhinotria secanalis. I'm butchering the Latin, but
Japanese knotweed.
yeah, exactly. It's simpler. So people like that. I was expecting a lot by Kimrex Gold, T3 extract, but some people have a reservation, might have metals. This needs to be investigated. I'm not too sure to the T3 will produce an aroma in the cannabis. I haven't been able to. Now I'm just talking about approved cannabis products for in Canada.
Well, you know,
the States, they have a lot more choice of products they can use.
The thing is that, you know, I don't know how many times I'm hit up with people in flower that have, you know, infestations or outbreaks, and it's like, there's nothing you can do, especially if you're getting tested, you're screwed. But if you can actually do that almost to the end, damn, you got, again, gold bars, man. Freaking huge information.
And talking about like beneficial that can be sold in another country, one of our competitors, they have a mite that we cannot sell in Canada, prone mite, and this mite eats russet mites and powder mildew spores. So this mite eats spores and it eats russet mite, 2 big problems in cannabis. So we're working. We hope that we can find this type of mite in Canada and be able to bring it to people. But
you want me to bring some up with me?
And so indoors and in greenhouse is a bit more difficult to achieve. Some people, I'm not a fan at all about sulfur. Some people want to use it. Some people are using it, and it's still allowed in organic too, I think I have. Yeah. And so sulfur and cannabis, I think they don't mix well. But some people, and it's never vented the way it's supposed to be for people's health, going back to work with the plants. And I've seen 2 incidents that the burner just went on and on and on until the toxicity was reached, and all beautiful room of flowers turn yellow and crispy in the night. And the anesthetist, though, resists. Sulfur will kill everything, good or bad. Some aphids will resist, and the anesthetist, the crazy mite, will resist to the sulfur. So that's another advantage of Anatis baccarum, the crazy mite. It resists the sulfur. So for prevention, for powder mildew, I would go with these peroxides or Sanidate or Zerotol, whatever, never on the plant itself. On the stems, around the plant, everywhere there's spores that have fallen. I clean and sanitize around it as much as possible. A spore itself, without the living oil, will live 2 weeks maximum. People think spores live a long time. They will live a long time in a tiny bit of plant material, though, a little tiny bit of leaf you haven't seen in the corner there, or a little tiny stem under a table. It will survive there as a cleocystes, another structure, like a fungi structure that will survive time. But usually powder mildew, the 3 strains that affect cannabis, the 3 species, I mean, they need a living tissue to survive. So unless you always have planned to inoculate them, if you start anew, you're not supposed to get, and it's not supposed to be, I'm looking for my term. It's always on the surface of the plant, the powder mildew. It's not inside. I mean, how do you call that?
Systemic? Are you saying it's not systemic?
Yeah, you believe it's systemic or not?
I personally believe it's systemic.
Okay.
Over time.
Yeah.
Okay. It takes time to become systemic, in my opinion.
Yes.
Yeah.
I believe that I'm more like that school of thought that would become, like, at first it's just very surface thing, but so
so I'm getting a list here. You're recommending perhaps do a dip with a Sulfoil X. Then eventually you might use a Xeritol to clean sanitation wise.
Mm-hmm.
What's the next thing? Is that the garlic barrier with cyclone, or sorry, influence with cyclone?
Yes, and also it's not registered for that, but our Bioceres CC too, since the Beauvaria bassiana is an antagonist of powder mildew and botrytis, and also the oil emulsificates the spores, because we got organic sunflower oil in our mix. We got no limit of application. We're certified organic, and there is no phytotoxicity with the vegetable oil present in the mix of Bioceres CC. So that would be, but it's non-official. I wouldn't combat just powder mildew with that, but that's a pleasant side effect, you could say, about the product. I heard good things. If you want to use some bacillus and everything like that, you need to to inoculate your plants early. It never works once it's settled, once it's happening. It doesn't have much effect. Dr. Punjab, Zamir doesn't like that much IPM or actinove.
Sure.
He said we're not fast enough to colonize, but as a prevention.
Sorry, is that a Bacillus pumilis?
Yeah, and subtilis too. The one in the Serenade, the one in the actinove or IPM.
Okay.
The name of the product.
Mm-hmm.
This colonizes the plants too slow. I mean, for it to act as a curative kind of thing. For sure, potassium bicarbonate is a good thing too, in a way. It has 2 mode of actions, desiccates them. This I could use, but it's a kind of harsh too on the plant. Could have some phytotoxicity. We even have some ornamental growers that were buying it to combat mealybugs. So with the potassium bicarbonate, so with the fungicide, they would So they would use it as a test to get rid of pests. For powder mildew, in fact, you try to have resistant strain when possible. Afghani descent is always the best bet. Some sativas do have resistance. You control your environment. Try to get the most even temperature day and night. You don't want any dew point happening on the leaf surface when you close these high pressure sodium lamps or metal halide lamps, and then it creates a condition that's perfect for powder mildew to spread like wildfire. So that doesn't help. Cleanliness, always making sure that's it. You can use you sterilize everything with the powder, with the peroxide, or with some alcohol on your gloves, whatever. Like, you try to always make it more. I would go maybe with UVC filters too. That would help too. They have wands in Europe they're selling, but you have to be very careful how to use them. They have to be at a certain height of your plants. You don't want to cause any mutation in your plants or hurt them in a way with those UVC rays.
There's I don't know, Leighton, if you've come across this as well, but there's a far UVC, which is supposedly not even toxic to humans, right? And so you can go in there with a wand and use a far UVC, and it's supposed to have a lot of the same properties as a pathogen.
No, I haven't heard about this. It's a wand that plugs into a 120 outlet that you just shine.
Yeah, you can walk over it. You know, they're battery operated things. Sterile Ray comes out with it. A company called Sterile Ray, and they also have like in duct units and stuff like that. So, yeah, I'd love to learn more about that myself. But that is just a safer way of using UVC. You can actually be present.
It has to be happening also when it's dark.
Oh, so you have to go in there after the lights are off.
Yeah, exactly. You have to apply the UVC. I mean, a certain device, they're applied when the lights are out.
Man, that guy's fast. He deserves a gold bar himself.
Or at least
thank you very much.
Wow, this is really interesting. Well, I'm gonna definitely read up more on that one. That's really cool. So, yeah, go ahead, Ab.
Oh, sorry. And you just mentioned Zamir Punja from Simon Fraser University, one of probably definitely Canada's leading researcher when it comes to cannabis pathology. He's done a lot of work with quite recently on hop latent viroid, and in fact, I think has a webinar coming up on 420 on hop latent as well.
Exactly, this Friday. Yeah, this Friday, 420. That's it.
On Thursday, yeah.
Sorry, Thursday. So,
what are your thoughts around hop latent, and in terms of perhaps some of the cultivation things that we can be doing to mitigate the spread?
It is really bad. It's like the AIDS COVID of cannabis, I would call it. It's really, 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 really bad because a lot of people don't know they're spreading it. It could even be caught from dried weed. There's going to be some virus present on the dried flowers. It could go through the seed barrier. This really scared me. The hope, though, is that with meristem tissue culture, we can save our strains.
Now, I've also heard that tissue culture can only reduce the inoculum.
No, but you have to do a work because it's the same thing in raspberries. We're doing it in raspberries. I was working on raspberry viruses before, collecting different ones with the researchers in Quebec about it. And you have to. It's a race with the viroids. So you do your first tissue culture thing, and then from that you test it then okay, then it's there. Okay, it's in the back, it's running in the back, but then you have to speed up, then you catch up, the new cells, on top. Then then you can. It is possible not to do it and to save with tissue culture, but it's a race, to these viroids are really bad, and the thing is also alcohol doesn't kill them.
Mm-hmm.
So you have to use hypochloric acid or this Furcon or Shield or these specialized disinfectant.
Sanitizer, disinfectants,
yeah. Exactly. So good old Javel water will work, but so that's it. You have to sterilize everything. And you bringing a new strain, you test its roots, you test its leaf, you test its stems.
Yeah, I think that's probably the most important thing with living soil bed growers, right? That's one of my biggest fears as I try to promote more of the living soil beds. I worry that if we do get hop Leighton, hard to really remove a lot of those roots, right? So, we have to do the quarantine properly, and we have to be very careful on what we're bringing into our
So, Claude, have you heard of the Earth Box? So Alan Adkins, this cat from NorCal up in the Humble area, did some work. He came from the dairy industry, and he was trying to deal with excess odors, neighbors complaining, started playing around with EMs and different lactic acid bacillus strains, and came up with a concoction of anaerobic soil conditions, which he demonstrated that you could actually take a plant with this virus, plug it into the system, and you had to be outdoor in sun. You couldn't be doing this in the greenhouse, but you put the box together, you put the cutting or the mother, whatever, into this Earth Box, and you left it outside for the season, ran it all the way through, and it would begin to produce productive plants. Now, they did not kill the virus totally, but they knocked it back far enough that the mother's clones would produce flower again, which was the main concern, is that it got to a point where it wouldn't produce anything other than vegetative growth. So if what you're saying is true, it's a race against time. This combination may be the key answer, is that you go ahead and plant this clone or mother into this anaerobic condition, and then you run it out and try to type the tops off as quick as you can, like you said, to chase to the new tissue or to the newest tissue, because obviously if it goes back to budding, then there's going to have to be some or a significant reduction in the virus during this process. So I highly suggest you look into that. Do you have leaf tissue culture guys up there that you work with?
Yeah, there's quite a few tissue culture companies that are offering that service, especially around hop Leighton. But but what I'm understanding for this is like essentially using nature to clean up a plant.
Yes.
And so you take a clone, and then as soon as you can take another clone from that clone, you're just reducing the inoculum that way.
Mm-hmm. Yeah, that's one way. But the interesting thing was that he could take a full dudded plant, not producing buds anymore, plug it into the system, and in the end of the season in fall, it was producing buds. So yeah, there's a combination, like maybe you're pulling a clone just before budding cycle, or you pull a bud and reverse it and re-veg it and then take that off and then scrub it. So I think there's a couple different approaches to it.
I mean, I like Claude's analogy of HIV. Because I often say, hop Leighton is very much like HIV or herpes, where it can be incredibly latent. And what you're really trying to do is try to make sure that your immune system is is top notch, whenever it decreases, that's when it's going to probably express itself. And so arguably your immune system is more aware, heightened through a living soil system. And so you might be able to suppress it, make it more latent. Yeah, I still don't know if we ever become HIV free or herpes free. We just become more latent about it. Yeah.
I don't know. I still like the hops latent viroid. Yeah, that latent viroid. Yeah, that.
Fuck you, Ken. I was waiting for someone to fucking throw that out there. Hidden, not latent. Hidden.
So, that's it. And that viroid, like in hops, only a few cultivars of cannabis, they show the symptoms. So that implies that both the symptom expression and disease severity are really cultivar genotype dependent. So it's the same in raspberries. We had strains that we didn't even know had viruses. And then this farmer would plant a new strain, a new variety, Boyne strawberries or Pathfinder strawberries, raspberries. And then we would get what is happening to our Pathfinders, And then we thought it was the Pathfinders that had something. No, it was the row just next to it that was spreading that virus to another the cultivar that was showing the symptom,
As far as we know, I don't think it is transmitted by insects, but
yes,
it is already.
Yeah, they haven't been. It's really hard to prove, but I mean, if they could do it, I'm sure. If a human touches it and transports a big bug, any bug, the viroid is so small, it'll cling on because already trips are transmitting viruses.
Mm-hmm.
So it must be transmitted viroids.
Yeah,
it's like really.
That's one of the biggest challenges with the ebb and flood benches right now, right? Because of course the inoculum seems to be quite present in the roots, and so of course if you're using a ebb and flood bench, you're just kind of mixing fluids and just spreading them around.
So in fact, with the viroid, in fact, you have no choice. Once the plants are received, 30-day quarantine is critical in order to properly evaluate for the plants the presence of plant pathogens.
It also, I think, now that we think it's seed transmissible as well. It's important that when we're doing a pheno hunt, and if you just get some new seeds, that your pheno hunt is done in a separate tent or.
So the Great Gardener guy was going on social media lately. Only old seed stock will be good soon. It's like, no, doomsday prophet. So.
Well, I think there's a lot of merit to making sure that we are somebody's got to take the diligence up.
Yes, exactly.
We have to be responsible because
exactly. But that's it. But it's especially this type of thing that's happening. There's no chemical treatment for it, but the meristem tip culture, thermotherapy, and cold treatment too. These have been found to reduce the viroid titer, but that's it. So you want to reduce the viroid titer. So it's really like if you combine and also the cold treatment and the meristem culture of tips, this will be even more effective. But
so talk a little bit about the cold treatment. Is that something like freezing, or is it
just incubating plant material 2 to 4 degrees Celsius enough in the dark for 8 to 21 months? If you have the patience. So that's the way to save certain strains of that's the way they found it. Thermotherapy, though, it's easier. It's 36 Celsius for 14 days. But it will decrease your titer of a viroid They call it. I don't know if I have it right, the name.
Mhm.
But the original titter will be restored after 6 months under field conditions. So he's like he's just like manage it in a way. So just to really like the cold and heat treatment, they could really potentially reduce the titer of HLVD, but subsequent meristem tissues they're required to completely eliminate all of the viroid. So and this knowledge, I'm taking it from this paper that was published last month, 30 minutes away from my place. It's Dr. Perrault and Dr. Adkar Purushotama. I hope I'm not butchering his name, from University of Sherbrooke. They did a paper on the hip latent viroid and the cannabis industry. So I just can't wait to see and to hear Zemir on Thursday. So anybody can register. It's on. You have the link. You can find the link on LinkedIn. It's Niagara College, and also the Minister of Agriculture of Ontario. That they are supporting this show. This not would be possible in our far right fascist province of Quebec.
He said it.
Oh my God. We have it's called Ligue d'eau in French. In Quebec, I used to work with them. MAPAC, that's the Minister of Agriculture of Quebec. And the Union of Agricultural Producers, UPA, is the big syndicate that's pushing GMOs like big time, thing like that. These two guys, they're so anti-cannabis. And Ligue d'eau is the only one. They're really nice people. They're like Vineland, I talked to you about, the research station. They're like marketing, they support all their ornamental growers in Quebec. And they have an agronomist that just helps cannabis growers. So if anybody needs an agronomist, besides my good friend Av, there's another one I know that just works with cannabis growers from Ligue d'eau. That's an institute, but there's a Niagara College graduate. It's a really nice school, and Sébastien Jacob is an amazing teacher. He's so intense and energetic. You must never sleep in his class, that's for sure. And, they have a cannabis program there to teach, and they were able to find there that for the first time that Anatis Bioprotection was able to reproduce in cannabis. So these crazy mites, they will lay their eggs in your substrate or under the table or the cryptic locations. They will, the female lives for about 3 weeks, and it will lay a little mass of beige reddish eggs, little clusters, 5 to 40 eggs, and these will hatch. And the first stage has only 6 legs, and when they come out, it's a bit creepy. They look like little hands coming out of the legs. And then the first stage already eats. A couple of days, builds a chrysalid. Then it's the first stage where it has 8 legs. Then another chrysalid, second stage. Then the third stage, there was a primal nymph, deutonymph, tritonymph, and then a tritonymph. It's like when they're teenagers, in fact. And then they go into chrysalid before they hatch as an adult. So when you receive them home, when we ship them, they're in wood shavings and wood bran, wood sawdust, and they it's full of chrysalid of teenagers that are going to hatch and adults that already emerge. They're running everywhere. So you get the latest stage, and some of the teenagers are coming up. So you leave your if you want to spread it in your garden, you leave your wood shavings in the bag or in the container, and you leave it in like that, and they will come out in a couple of days. But the adults will have left the container that same day. So it's fascinating, all these different things.
I'm starting to get a
Claude,
back.
Sorry, Claude. Let me ask you this: do they have a natural balance or correction, or will they just go crazy and overpopulate? And
they will cannibalize each other if they can eat it. They'll die of hunger, or cannibalize, or wander off, or die of cannibalize each other if they can catch each other.
Wow, that's fantastic. So they have a great corrector built in.
Yes, exactly. So because no, they haven't. And it's funny, I did the Grow Up show in Victoria, and that morning we had a booth there, and we were expecting it with a courier by plane or anything. So that morning, the expo was starting. I didn't have my booth yet, and I just finally had received a bag of crazy mites from our guys in North Sandwich on Vancouver Island from Applied Biodynamics. And then I was talking with FedEx on the phone, and I was looking around me, and it was the smoking lounge, outdoors consumption area, and all these nice people were enjoying cannabis, and it was beautiful weather under this tent in Victoria City next to the Grow Up Expo. So I said, "Oh my God, I'm in hell. I'm talking to hell in paradise, you know." I'm in paradise talking to hell. Then I look over my shoulder, I look at the ramp, and the concrete there. Crazy mites everywhere. They're all the native ones are there. They're all running everywhere. And you see them usually in very hot days in summer. They're cosmopolitan, so they're all around the North Hemisphere. And I think there are a couple places too in the South Hemisphere. If it's not them, it's close cousin. They're really on their, they have their genera on their own. I mean, the Anatis, that's really a type of of mite, a predator mite. So
Yeah, that's a nice little opportunity for me to plug the Grow Up conference. Claude's going to be speaking at the Grow Up conference. I think he's going to be hosting a couple of panels on some of the novel things that are happening in the IPM world, but also going to give a talk around integrated pest management as well. Of course, Leighton's going to be speaking on the horizontal living bed system. And, I should also mention Claude's one of our annual judges around the cannabis flower competition. And the I think he's going to do hash and concentrates as well.
Yes.
It's usually back to back.
He's showing my beautiful Friesland indica.
Is this your backyard?
Yeah, he's playing in my Instagram. This was the Friesland indica, circa 1989, from Super Sativa Seed Club.
Nice.
And it was one of the two seed banks that would ship to America from the Netherlands. One was Neville Schoenmaker, the great Neville. Yep. Rest in power. He left us a couple of years ago from cancer. He flew to exile into Australia in 1990 when the Operation Green Merchant, or another one from the DEA, happened. They followed 1,000 shipment from the Netherlands to showing my sublimator. And my first rosin press, before the fancy rosin press, were the t-shirt press.
Exactly, or an iron.
Or the hair fucking press, the hair straightener.
Exactly. This was the first. The work, but it was so waxy, and the taste wasn't there yet. We haven't found the right temperature and the right pressing. This was nice though. This was a CO2 extraction of Tangy. It was so tasty, like pure orange. Pure orange it was done with a supercritical CO2 extraction. Such an expensive machine. It's such a tedious machine to work, but it did wonders. One part turned solid and one part turned liquid. And the part that turned liquid was so liquid, we could put it in carts. Even the non-fancy, the first generation carts, and it would work so well. Didn't have to mix in any of these.
You must have done that as a subcritical as well, then.
Mm-hmm.
I mean, unless the terpene survived supercritical, wouldn't it?
Yeah, sorry, not the supercritical CO2, but the subcritical. I mean, to preserve the taste,
Yeah.
the essence of the, and it was so intense. It was the first tangy on the market, the first from a DNA or Crockett Family Farms, I think. The first like real super lemon terpenes. Really, he was showing some samples of the first cannabis cups I did in Amsterdam.
Yeah, we'll have Claude judging some flower.
We had to do the coffee shop hop, so we had to go. We didn't receive all our samples at once, all easy, and it's like but it was fun, We had to run around Amsterdam and trying to find where the coffee shops are, to find each entry to get back and to smoke them where we could to judge them, and then go to the big expo they did at times. I was there one year. Times did a big mistake. They rented a space in the suburbs of Amsterdam, in a very conservative suburb that's really anti-cannabis. So it wasn't a couple hours it had started. Mila was there, the hash queen, the guys from Greenhouse Seeds, Franco, rest in power too, was there with this giant vaporizer, with the big the volcano with the big balloons, and you fill up with you would vaporize an ounce in the big balloon and give it to everybody to enjoy a vapor of a super lemonade or something like that. And then it was going on, and then one of the iTimes guys started to say, "The pineapple is very good today. The pineapple is very good today." That was a single. There was cops in the place, so they started to patrol everywhere. And then the first thing we learned, the place was surrounded. So they busted the whole areas, like the expo scene. So people started to smoke everything they had, everything they had, as fast as they could, because they were starting to empty the place of people, and you had to go through this corridor and empty your pockets into this big garbage can. Nobody got arrested, just Mila, the hash queen. She got arrested, maybe because she protested or something like that. And they released her that same night. But anyways, and there was these guys from NorCal Seeds, I think, or North, anyways, and they had this big chocolate fondue, and they were giving to people a big container of a hot chocolate fondue that was infused big time. And they were deep shit because they had to eat everything, because in Amsterdam, in the Netherlands, if it's not solid at room temperature, it's not legal. So you cannot have oil, you could have rosin, you could have something that's solid, you could have ash, but it falls under the opium law when it's a liquid. So when it's liquid, so this counted as something liquid. It didn't count as a space cake or something like that they serve in coffee shops already, So the guys, they must have been so stoned, they ate everything. There was enough for 100 people, and just a couple of guys they drank and got rid of it too, I think, also. But yeah. He just had a memory flashback because of Ken playing in my Instagram. About this kind of Amsterdam. I miss the city of pleasures, all pleasures. Incredible. You meet so many people from everywhere to enjoying a good smoke. I wish they have this consumption lounge. And anybody hits Vancouver, I advise them to find out about the book club. It's a really nice place. It's a social club. It's a nice place. Events happen. You can smoke and share with everyone. It's a really nice place, and they did it in a way that it's legal. So and it's also some research stations now Lit in Toronto, I think it's called. It's a place where you can go, and the licensed producer, they can even gift samples to people to try out their adults that show up. You can test, do some scientific research on the olfactory and gustatory.
All right, time to get you back on track, Claude. Here, what's this picture, man? We'll bring this up for you.
This was, I was working for greenhouse research. So these are greenhouse peppers. These are from the Ramiro type. So they're long and sweet, very, very sweet, very tasty, but not hot at all. Very, very sweet. So we were trying two different things. It was almost 20 years ago. The first LEDs, we were testing them with peppers to try to improve our crops. Back then, we only had single colors. Didn't have these fancy. It went so fast, the science of LEDs, and it's amazing the leaps and bounds that it took. And so yeah, these was Ramiro peppers. So we're doing cucumbers, different types of tomatoes, and everything. I had 3 greenhouses to take care of. It was a lot of fun. With the harvest, weigh the vegetables, and do all sorts of things.
That was a really nice dip into the subculture. So I love hearing those stories from the 80s. I was wondering, should we jump to some questions or?
Yes.
Yeah. Leighton, do you have any right now, or should we go to?
No, I was trying to formulate. There was one that jumped out at me almost back in the beginning, and now I had it and then I lost it. So why don't we hit on the questions that we have, and hopefully it'll pop back into my head.
Okay, well, we'll start with Silly Lily. You were talking about some of the powders, and she was asking Claude, would DE work?
The item assert is fossilized exoskeletons. It can work if it's a food grade size, it's fine enough. Then it could be even used as a substrate to carry nematodes. But anything bigger than that, the item assert will, like your predator mites, don't work with it. It will desiccate them. It will be like tiny little razor blades for them, and it will hurt them much. Yes, especially as a barrier, item assert would work a bit on young aphids. Pretty sure it would help. I never thought about it, but it's a good another tool, I think. I used to use when you have a rice root aphid, which I didn't talk about. Bovaria is amazing in the root system. You can drench your sprench when you're not allowed your plants with it, and it really kills them all. The rice root aphid, because they're another big threat now in cannabis, and then rice plants and ornamental grasses and lettuces too, they attack. So these rice root aphid with the drench from Bovaria, and I would do these rice root aphid also go up and down the stem of cannabis. They can even go up on the top of plants sometimes, and we would get them with tangle foot tape that we place around the stems. So the ones that go up and down, we'd get them, and the ones that go up, they get parasitized by the Ephidius materiae, or they get eaten by the Chrysopa larvae, the green Chrysopa or the brown Chrysopa, the lacewings, the brown lacewing and the green lacewing. So we have the brown lacewing too, which is different than the other green lacewing, meaning like lacewing usually the adults will only eat pollen and nectar and whatever. So they're hard to have a cycle in a cannabis greenhouse, but the brown lacewing, they will eat meat too. So they will go after, the adults will go also after aphids. So they don't rely on their own pollen, so they can cycle in a cannabis greenhouse.
Fantastic. So the next one, we were talking about the UV lights and how long do you leave the UV light on? How often, and again, how long?
For air filtration thing, I think it's on all the time. If you're doing a one over the plants, I'd have to check. I don't want to say anything that could be wrong, and I like to be precise in my answers. It has to be just a certain time. Like you cannot. You're going to damage your plant if you just put it over your plants and leave it on. It won't help them. It has to go. There's a certain speed, but I want to be sure of what I'm saying. There's a certain speed that you go over it with your with it over your plants that you have to respect for it to be efficient and to not damage your plants. Like UV rays will damage your genetically.
And what about like the underside of the plants and stuff? Like, do you have to go under and over, or use a fan to flip the plant around? I mean, I'm assuming you have to expose all sides of it, correct?
By people that do it usually do the top. I mean, meaning it depends on dense your canopy is too. They'll make it more efficient or not. It's the speed of what you're placing that's going over your plants. It's not. I mean, it's not. It's still in its infancy. This method, in fact, there's still methods and things to be improved. Like I know the big guys in greenhouses now, they have these robots that go over their plants during the night. So they're all programmed with the right speed and the right intensity and the right. So and to be 100% sure, I need to do some more research. I prefer to say exact things.
Yeah, we don't want bro science, Claude. We want the facts. So I think somebody should go to the website that we had up earlier, because I think they had a lot more of that information on that website as well. So
yes, exactly. The one you showed, the one and everything. Yes, they will have.
Yeah.
It's available.
Oh, sorry. Silly Lily wanted to know, Claude, what was the name again of that club in Vancouver?
It's a book club. It's over Burb, which is a legal cannabis store in somewhere in the suburbs of Vancouver. I forgot exactly where it is, but it's one of the locations of Burbs. So you go through, you go in the licensed store, and I think it costs you $10 to spend the day there. Because what happened when I went to grow up, Lyft in Vancouver, I had programmed, I don't know why, my agenda to catch my flight back to Montreal with the Montreal time. But I was 3 hours behind. So I was in my cab going to the airport and look it up. I said, oh no, what did I do? But I think it's my subconscious that made me do that, because it's like some people had invited me to the opening of that social club, the book club, and I said, oh, I cannot, I'm leaving tomorrow morning, I have to catch my plane. Finally, I was able to make it. So I spent my whole day there with amazing people. There was food, there was drinks, there was music all day. It was really, really nice. A lot of people came and shared a lot of cannabis. I met Che from Rosebud Farms and Unicorn Cup, and it was a charming lady. I forgot her name. She was there, his partner, and these people from Noble Cannabis, the Next Friday brand, which is the one that's the most has the best reputation on the legal market these days, and it was a lot of nice people, and it is Adolfo that started that with the help also of Axel, French expatriate, a very nice guy. Every time I meet these guys at expos, I always have a lot of fun. So they started that, and Vancouver is really nice. It's happening every weekend. There's special events. I'm sure they're open for 420. So it's not it's not an everyday thing, I think, but I mean it's a weekly thing. It's a weekly celebration, that's for sure. Yeah, DJs, musicians, performers.
Yeah, Hillbilly wanted to talk about the one in Toronto.
It's called LITE, L-I-T-E. So if you do a search, I'm pretty sure you will find it. LITE research, LITE cannabis research. I'm doing the research.
Both searching.
It's LIT, L-E-T, L-I-T, not no E. Yeah, LIT research, consumer-focused cannabis research. So you're able to be available for, it's a cannabis tasting experience. So in fact, you have to be 19 or plus in Ontario. And so you can socialize with people there. And there's all sorts of sponsors, all these licensed producers that want to know what people think about the product. So Pure Sun Farm, Sundial, Montreal Cannabis, 5 Point Cannabis, Simply Bear, all these people are there. And I mean, they will supply certain weeks, certain samples for people to try. And people, like in the Canadian law, they're allowed to carry an ounce with them to the research site and to share with people. So you can share with your people some your cannabis and enjoy the one that's been provided by the licensed producer for the public to judge and test, to do some research and reporting for development of specific medicines for certain elements and taste. And so we have to find our way across their laws, make it happen until they finally, they're the first lounges that were accepted in Vegas. But it was just a matter of time for them. Some real studies going on. Volcano Station, right? Exactly, 30 samples. Yes. It's all part of the research.
So I'm out of questions, so you guys can take back over.
So let me ask you this: Is Lyft still a thing up there in Canada?
Yeah, it's owned by a U.S. company, and they still are planning a Lyft Toronto, June 1st to the 3rd.
Yeah, exactly. And they have one in San Francisco too.
Oh, are they? Okay.
Yeah, and they just had one in Vancouver, but they're MCI USA. So it's not the me, I love Grow Up, because Grow Up is like a activist, volunteer, interesting, real. It's not just the business side; it's the real activist.
Well, that's what I was going to ask next. I was under the impression that Lyft was really about the money boys playing money.
Exactly. It's all they call them the Chad.
Chads and Tads or whatever it is.
Playing them with the money. Like building billion-dollar companies and bringing them to the ground, producing billions of grams they have to destroy. Anyways, I'll stop there. But Grow Up, just an example. When I have a booth at Grow Up with a company, they give me 10 passes for my customer. They want us to speak. It's an advantage. It's a value for them. But at Lyft, they'll charge me if I want to speak on their stage to bring them value to their expo. And they will also won't give any pass to anybody. So you have to pay for your pass. So in June, I'm moving the first week of June, and it's happening the first week of June. So I think my team member, Stephanie, will be there. Stephanie Knox. She'll be going on the floor trying to meet people there. But we won't have a booth. We have one at Grow Up in the May.
Well, we love you for that. That's great. Yeah. We look forward to it.
Yeah, Grow Up.
And that's what Grow Up's all about. It's a lot of a lot of expert to expert talking, and so it's just going to be a lot of.
It's not just the commercial. It's the cannabis, the seed exchange. The seed exchange is amazing too. People trade genetics.
Yeah, it's kind of getting back to the old shakedowns at the dead shows.
So all the OJ of growing and the newcomers meet up to have a lunch together, cultivators luncheon, and And there's always the consumption lounge, very important place in the expo, because it's all about the cannabis. Because these lifting, they used to have no consumption lounge, whatever, and you go after at the after-hour party. You cannot smoke cannabis. It's all people with drinks, getting high on alcohol, and what? It's like cannabis show here, like so no lift.
Now, I did have one question. I don't know if Leighton remembered his question yet, but this is a question I was going to ask you. Sorry to interrupt and go back to IPM, but I was just wondering, are there some foliars that you really want to avoid spraying if you've already released benefits?
If you release beneficial, if you use,
I like that. And I just want to comment on Mad Dog's comment. Obviously, Mad Dog is in the UK and he's ready to celebrate 420. So.
Okay. Thanks. Yes. Oh yeah, 420 day. Anyway, so yes, for sprays, Bavaria only. I prefer to use it during propagation, during vegetative. I would never pass the 3rd week of flowering, because if you have to test your things, a COA, they might mistake the Bavaria as a nasty fungus. They don't differentiate. So that would be careful with that. Anything that damages trichomes, if you can use a cold fogger, it's always the best. Cold fogger does the best application without dripping, especially for Bavaria. You don't want, it's the best way to spray Bavaria is with a cold fogger. Be careful not to heat fogger, but a cold fogger. It will kill off the spores if there's a heat. And that way, it really does you use less product, and you don't miss a spot. And it doesn't. When you use Bavaria, you don't want it to drip off the plants. You want a good coverage, but you don't want it to drip off the plants. It makes it less efficient. Oils or soaps will kill all predatory mites. So if you have to use these, really, you use them before. Once the soap or the oil is dried up, then you can release your predator without any harm. Because if not, you're just throwing your money out. It doesn't worth it. You shouldn't use the predators if you really want to spray all the time. In Canada, you have to be careful. The only 4 products are approved, like 4 of the most used products are Copa, Opal, Nudisand, and Exile, and it's all the same thing. 47% of potassium salts of fatty acid. So after a while, it denatures the trichomes, I would say. I am not a big fan. For sure, it works. I hate the smell. When I come in the facility, I say, "Oh, you use the Opal, Copa." I say, "How do you know?" I know the smell. I hate that smell. So I'm not a big fan. And the certain company, I won't name the one, they're going around saying, "Oh, beneficial insects don't work. A good spraying program that's what works for IPM." So anyways, I'm not trying to start a war with them. It's like, for sure, soap could work, especially when you have to do a reset, like I would say. Like there's too many pests. Beneficial, you'd have to throw armies and armies and armies and armies, try to get over it. Then you can do a reset with an oil or a soap. You try not to be careful with the phytotoxicity also.
You don't want to mix the two.
Yeah, exactly. And the soap and all, they can like clog the stomata sometimes too. So you want to be careful with that. The fragile pistils, the bracteals too. The spray with the trichomes to don't want to knock them down. This is an overfly he's showing. We're still figuring it out. It's not easy to use this predator. This is the larvae of the overfly we've seen before. It doesn't look at all like it's an adult. This is pupa. It is the adult will eat nectar and pollen, so it needs flowers. So in cannabis, we're just growing females that have no nectar, no nothing, no pollen. Just once sporadically, there will be some pollen if we're using males to produce seeds. So cannabis facilities, I wasn't able to really test them yet, because these adults, they will lay their eggs next to the aphids, and their larvae are amazing predators of aphids. They will clean up colonies very fast, and they're like the worms in Doom, the movie Doom. They really look like crazy, like the way they grab an aphid and they chomp it down like vertically, like it's like crazy. The way they and they're so big. They I don't know. They must spread a pheromone that puts the aphid to sleep, because the aphid never panics when they're there. They're like furtive. They're like so big, and then they're just there and inside the colony, chomping down on their little sisters, and aphids like the other aphids don't budge, don't move.
So I did remember my question, and I think you've just already clarified it. There is no soil phase for those candidate aphids. Everything's up in the canopy, correct?
Yeah, meaning if the eggs haven't been laid in the ground, this will.
So they do lay in the soil as well?
Yeah. Aphid, cannabis aphids, yeah.
And so is there a predatory nematode that will work on that life cycle, or the eggs that are in the soil?
There's still, I'm pretty sure that our predator mites that eat any invertebrate eggs in soil will help to clean that. And maybe the rove beetle too. But it's not something that's been studied yet because of the lack of cannabis studies in general, and the lack even more of cannabis aphid studies, because it's also new and nobody has wanted to put the money in it, or at the time to do it. Very limited researchers, not many people doing the work. So yeah, you never. It's the lack of that sort of research in this field. But we try to palliate the best as we can on a smaller scale what we can do to associate ourselves. Like now I know that I have a good method to really start my plants clean when I have cannabis aphid with the Bovaria, and not just pushing our strain too. It works also with the competitor strain, the Botanigar, but Bioceres is more virulent for cannabis aphid, according to one researcher yet in Canada. So and a blend of Bovaria would be even better, I would say. Use 2 strains would work even better. But once you do it during propagation, because you spray it on your cuttings, and it's so humid, if you're doing it under domes or under curtains, it's always 80 to 100% almost relative humidity. The Bovaria catches like crazy, that we even see after 8 or 9 days, we see the final stages that a lot of time we don't see.
That to me is worth another gold bar.
The fructification of the cordyceps, like we really see them as fuzzy white balls. Like we can count them, the aphids that were on the stems. Oh, there was one there, there was one there, there was one there.
That's I mean, that's a great thing to do during that time.
This is showing the midge. The orange predator was the aphid predator image. Anatis Bioprotection, the one that I prefer using for outdoors, because it's so cheap to use outdoors, and you're just boosting their natural populations. And you don't have to put 1,000 per acre. You put 250, because they will not help each other if you put too many at the same time somewhere. And certain companies that want to sell a lot of things, they're not happy with that, what I'm saying. But if you use less, it's better. Just 250 per acre is more than enough. So that was the adult midge we saw. And it's amazing how it can, like, it doesn't stick in the spider mite, not the spider mite, but the spider's webs, different species.
We've hit our 2-hour mark, and you know, I would invite Claude to panels and give him 15 minutes, right? So what an incredible disservice. He's essentially just giving us a 2-hour crash course on IPM. It's absolutely phenomenal. I think Silly Lily said it all in her comments, just about grateful for the amount of knowledge you've just the gold bars. I think there could have been a few more. Not to blame Ken, but he was being stingy with the gold bars today. But
what? I'm trying to bring up websites, and you want gold, okay? Claude was giving you the gold, baby.
But I'm gonna just call them teasers. Hopefully, folks can join us in Edmonton. Eventually, I'll get a discount code to the Grow Up Conference and get it out to you. So. But looking forward to that, and just want to give thanks for that.
And I'm sure there was a lot more that he could have dropped because it's only 2 hours.
I did a presentation for agronomists in Quebec a couple of months ago, and I talked for 2 hours and a half just about trips.
Yep.
It's fun.
Well, we'll definitely have you back on, Claude. I definitely want to hear more about the work that you're doing and these these new crazy bugs that are just like freaking Superman.
Yeah, you're gonna take some home with you from Edmonton.
Yeah. I thought, I'm bringing some of those home. What did you guys want me to bring up?
Mike eats the russet mite.
Okay.
Yes.
And fish brew.
We're testing right now our Biocerus CC against russet mites, empress and mites, to see if it works. The oil will have an effect. Some Bovaria might affect. So I hope, because Matthew Gates, one of my fellow bug enthusiasts, that's an amazing contributor on the Future Cannabis Project channel. He did shows with me before. He has his own channel, Zentanol Consulting. He's an amazing entomologist from California, a young guy, much younger guy than me, but he has many years experience. Anyways, and he was talking about that, the Bovaria and the russet mite too. It could be controlled a bit by phallacis, but not much. It's really the oils that will get them, the empress and mite. But we're testing right now the cyclamen mite, which is a close cousin to the empress and mite and the tomato russet mite, with the crazy mite. But we don't have much hope.
It's like the Energizer Bunny. You just wind them up with one question.
But we think it prefers larger prey, so we might not have success with it. We'll see. I'll keep you posted.
Sounds great. Well, we look forward to having you back on in probably 4, 5, 6 months and get an update on all of this.
Great.
And as always, pleasure. Ken, thanks for all the work behind the scene. And Claude, thank you so much for giving us 2+ hours of your time to come on here and really help us, get the word out about some of these, beneficials that are so important for the future. So thank you again.
Yeah.
Thank you, guys. Hope to see you guys soon and in person in Edmonton too.
You bet. Okay, guys, with that.
So, Leighton, are you from Canada or from the States?
I'm down in California.
Oh, okay, because the crazy mite, that's the cannot ship, because I would have reserved a container for you. But you do what you have to do.
He's going to take some back in his hair.
Okay, guys. Thank you, everybody, for being on the chat. You guys are amazing. And for myself, I've got the 420 Cup coming up on the 19th, starting at 5 o'clock in the Amsterdam Cafe, which is another consumption lounge in Vancouver. So that's going on. And then, of course, the 420 Cups all around Vancouver. Everybody's going to have a hell of a good time for 420. Guys, thank you for our hosts, our guests. You guys are just amazing. I might be doing some work in the background, but I am learning constantly. Thank you guys so much.
Thank you.
Ciao. Peace out.
Hey, guys. Peace out.